this health threat. AMR is a complex issue that requires efforts from all professions, and not only those in the medical sector.
Say no to AMR.
This is why we are proud to introduce to you the AMR Ambassadors Program for young people in Africa. The program is open to all university or college students from any institution in Africa. Are you a student pursuing arts or creative sciences, building sciences, law, international relations, behavioral sciences, journalism, engineering, health sciences, IT, or computer sciences, accounting, business or economics, agriculture sciences, or any other course? This program is designed for you. Now, let's engage.
Hi guys, thank you very much for making it in time. So as we wait for a few more of our friends to join in, and as we wait for our speaker to join in, we'll play another video. But as our speaker joins in, please feel free to introduce yourselves in the chat room so that she gets a feel of the people who are here and where they're coming from, which professions they're pursuing.
What does the AMR Ambassadors Program for young people in Africa involve? The AMR Ambassadors Program is a 9-month program that seeks to 1. Raise awareness, 2. Improve understanding, 3. Promote sustainable engagement of students in universities and colleges in antimicrobial resistance. The program is divided into 3 phases. The selected students will have the opportunity to meet with and be mentored by experts from different professions hailing from different parts of the world. They will also interact and engage with other students from different countries in Africa. In phase 1, the successful applicants will interact and learn from each other through focused lectures, group discussions, workshops, and engaging with mentors. The sessions will focus on equipping the students with knowledge on AMR and helping them develop skills and practical creative interventions that can help them engage actively in the fight against antimicrobial resistance. In phase 2, participants engaging in teams of 3 to 5 members will learn more about design elements, system thinking, and project management. They will then be challenged to come up with a creative AMR intervention that showcases AMR as a multidisciplinary issue in small groups by leveraging the knowledge gained in phase 1 and concepts learned in their professional courses. Present their work to experts and revealing to organizations and experts who can support, refine, or implement their interventions beyond the program. In phase 3, participants engaging teams of 5 members from the same university will receive some funding to design and implement contextualized activities aimed at initiating or supporting sustainable AMR efforts within their local universities and the surrounding community. The best 2 projects will receive an award of $750 for the winning team and $500 for the first runner-ups in the final closing ceremony. All participants will be awarded with certificates. Visit our website to learn
So hello once again. Thank you very much for joining in. My name is Anastasia Seboa. And our speaker for the day has already joined in. So before she starts off, I'm just going to give a brief introduction. So Dr. Margaret Lubwama is a medical microbiologist and lecturer with Makerere University College of Health Sciences at the Department of Medical Microbiology. Her main research focuses on understanding the resistance mechanisms of bacteria. She also sits in the infection prevention and control antimicrobial stewardship committees at the Uganda Cancer Institute, where much of her work on antimicrobial resistance has been carried out. Due to the scarcity of new treatment options for resistant bacteria, Dr. Lubwama also studies about antimicrobial activity of herbs against resistant bacteria. The goal is to determine if the herbs used in Africa are an option for treatment for resistant organisms. As a mentor and lecturer, Dr. Lubwama is passionate about teaching the community about ways to prevent antimicrobial resistance with the hope that the community will embrace their role in the fight against antimicrobial resistance. Ladies and gentlemen, the speaker for today, our presenter, is Dr. Margaret Lubwama. So allow me to hand over to you, Dr. Maggie.
Thank you so much, Stacey. Can you hear me?
Yes, we can.
Okay. So I sent you an email via your Superbugs email address, just in case. Okay, so.
Okay.
Can you see my screen?
Yes, we can.
Which are you seeing it in PowerPoint form or in ordinary form?
In slide presentation form.
Okay, in slide presentation form. Okay, so we're good to go. Can you still see it?
Yes, Dr. Maggie.
Okay. So hello, my name is Dr. Margaret Lubwama again, and I'm going to take you through this talk, which is biology of microorganisms. I do not have any conflict of interest to declare. And this is the outline that I will follow. I'll give a brief introduction, history of microbiology, something about taxonomy. Stacey, can you still see my screen?
Yes, we can.
Okay, I just got a message that, okay. So biology of microorganisms, and I will use bacteria as an example. And then we will talk about, of course, the morphology, the growth cycle, and some aspects of the physiological requirements for growth. And then I will talk a little bit about other microorganisms, viruses and fungi, not so much in detail because 1 hour is not enough to discuss all this. And so I won't necessarily dive into this. I'm going to make this presentation with my video off because of my internet bandwidth, but at the end of the presentation, I'll put on my video so that you can see who's talking to you. Okay, so did you know that we are not alone in this world? And nope, I'm not talking about aliens. I would like you to read through this sentence and think about it. Inside your intestines, in your mouth, and on your skin, there reside millions and billions of microbial cells, 100-fold more than the number of cells that make up a human body. And that's fascinating. And sometimes it's important to really think about that. And what are they doing on us and what are they doing in us? We may not necessarily answer that question today, but then it's important to know that within you, there are other beings inside of you and on top of you. So on the left picture, you have normal small intestine, the normal small intestinal mucosa. And on the right, you have a picture of the skin. And floating above each of them, on top of each of them, is what we call the microbiota, the very many types of organisms that are seen above these structures, okay? And they are put there to protect the body. They are responsible for resistance to colonization by exogenous pathogenic microorganisms. That means if they are there, then the microorganisms that cause disease will not be able to stick around. And that's one of the dangers of using antibiotics. When you overuse, one of the dangers of overusing antibiotics. When you overuse antibiotics, you kill the normal flora. This is what we call the normal flora, sitting above these important structures. You kill those microorganisms and you give space for disease-causing microorganisms to thrive. Okay, so this, you have your intestines, you have your body, but within us there are other organisms, very tiny organisms, what we call the microorganisms. And because they're normally there, helping us out, we call them the normal flora. So looking at some definitions, what is a microorganism or a microbe? It's an organism that can be seen only through a microscope, as the name suggests. You cannot see it with your naked eye. And then terms like microbiota is an aggregation of microorganisms, including bacteria, archaea, fungi, protozoa, and viruses. And then going a little bit deeper, we have the microbiome, and this is the composition of all microbial genes in a community. And so you have the microbes or the microorganisms, and in order to see them, you need a microscope. That means you cannot see them with your naked eye, okay? You need the microscope to see them. So again, you have your small intestines and the skin with their microbiota, and that is an aggregation of the microorganisms. So when you study these organisms, and then when you study the genes in each of these organisms, and then you put it all together, then you get the microbiome. And so there are many studies that have looked at the different microbiomes of the different body structures. So for example, you have a microbiome of the small intestines, and you have microbiome of the human skin, etc., etc. Okay? So that's a brief introduction of microorganisms and the normal flora and how they are connected to us. But it's important to understand where microbiology came from. It's not a science that just appeared. It has a history, and it's seated on very many giants. And without their input, we would not be where we are. So where did microbiology come from? Infection and microbiology followed different strands of, during the century. So there's one group that was really focusing on the infection, but then there's another group that wanted to understand the cause of infection. And so this group dived into microbiology. So ideas of infection and epidemics were recorded by Hippocrates, but it was nearly 2,000 years after when Fracastro proposed his classic, The Seed of Contagion. Okay, and he believed that diseases were caused by seed-like entities which could multiply rapidly, propagate quickly, and were unique to each disease. So he didn't really find the cause, the organism, but his understanding is that there was something that was causing diseases to appear the way they did, and different seeds of contagion caused different diseases. And separately across the board, we had microscopists who began to make observations on objects too small to be seen by the naked eye. And here we have the Dutchman Anthony van Leeuwenhoek, who made a remarkable handheld microscope and found many microorganisms in materials such as water, mud, saliva, and he recognized them as living creatures, and he called them animalcules because he noticed that they swam about actively. And so this is his microscope that he created, that he used to view these, okay? And so before the discipline of microbiology really was established and began, there were other discoveries by other giants in microbiology, by other scientists. So for example, you have John Hunter. I think he did the most unimaginable thing. So he inoculated secretions from sores around a prostitute's genitals into his own penis and was able to demonstrate transmission of infection of syphilis and gonorrhea. Then you have people like Edward Jenner. I did not include him in this, but Edward Jenner was responsible for discovering the concept of vaccination, and he showed cowpox was effective and safer for the prevention of smallpox. Then you have people like John Snow, and he showed that by preventing access to water, which was linked to a cholera outbreak, further infections could be terminated. And then Ignaz Semmelweis showed that fatal infections in women following childbirth could be substantially reduced in those attending the birth if they applied hand hygiene, simple hygienic measures. And he is said to be the father of infection prevention and control. So these were observations made from infection, and the idea that these infections could be stopped by certain measures. They still had no proof that it was microorganisms causing what they observed in virus infections, but then they were able to understand that there's something going on here, and there's probably a cause which we need to, excuse me, which we need to stop in order to stop the illness. And then later we have two important figures, Louis Pasteur, who is on the microbiology side, but also Robert Koch, who played, and both of them played central roles in establishing the microbial causation of infectious disease. And I feel like Koch is placed here, but he also belongs here because he actually made the connection between infection and microorganisms, and he did it beautifully. So Louis Pasteur, for example, was the one who showed that it's the presence of living organisms that were essential for fermentation. Initially, it was thought that fermentation was purely a chemical process, that is fermentation responsible for alcohol formation. Initially, they thought it was a chemical process, but Louis Pasteur demonstrated that it's actually the presence of living microorganisms that was essential for this fermentation process. And then he also refuted what was called the spontaneous generation, which said living creatures could arise from non-living matter. And he showed this by showing that nutrient solutions remained sterile if microbes were excluded. So it's not necessarily that living creatures could arise from non-living matter. And then finally, we have Robert Koch. And again, he made a beautiful connection between infection and microorganisms. He established the techniques required to isolate and propagate pure cultures of specific bacteria in the lab. And through his processes, he was able to establish bacterial causes of anthrax, tuberculosis, cholera. And from this, he formed what is called the Cox Postulates. And there are 4 of them, and I'll just go through them quickly. So the first one is that the organism is demonstrable in every case of the disease. So if you have a disease, then you have an organism, and you should be able to demonstrate that you have an organism. And if there's an organism, then his second postulate was that it can be isolated and propagated in pure culture within the lab. Okay, so in vitro. And this organism that you have cultured, if you inoculate it in another person or in another animal, it should be able to reproduce the disease that it did at first. And from this second animal or from this second person, you should be able to isolate the organism and from this new host. And so those are his 4 postulates. And it seemed okay for that period of time, but then of course, there are various reasons, for various reasons, application of these postulates is impossible. Yes, Cox said that you should be able to demonstrate the organism in every case of disease. It should be able to be grown in the lab. You should be able to put it in another person and it causes the same disease. But then there are times when these postulates is not, when it's not possible to apply them. So not all the time is an organism responsible for an illness. For example, you have autoimmune diseases, you have cancers. It's not necessarily that there's an organism that's causing that. And Cox postulates were focusing on bacteria, but then there are other organisms such as viruses, and you may not necessarily put them in a culture with artificial media. Such organisms need need cell lines in which they can grow. And so over the years, there have been advances in technology that have got us to where we are now in microbiology. And what seemed as mythology and theories back then can actually be proved today. So we have a more precise understanding of the nature of microbes. And of course, one of the most important developments was the elucidation of the structure of the DNA. I hope you'll have an opportunity to do molecular studies in this course. If not, probably another course will be arranged for it. But just being able to look at the molecular structures, for example, the DNA, that gave us a huge leap forward and a move into molecular genetics. We're now able to use molecular explanations to understand the organisms in depth, and this transforms our insights into the ongoing evolution of the microbes that cause disease. And so, but where we are now would have been impossible had it not been for these giants in microbiology. And so I felt that it's worth mentioning some of the steps and the stages that were taken to get us to where we are in medical microbiology. So before we get into biology of microorganisms, it's important to understand taxonomy. What is taxonomy? So it is the science of naming, identifying, describing, and classifying microorganisms into evolutionary related groups. Of course, as the name suggests, or as the description suggests, it's composed of 3 components. Okay, so you have classification, you have nomenclature, and you have identification. And why is taxonomy important? You have so many organisms, and in order to clearly understand them and how they're connected to each other, you need to put them into their respective groups. And if you can categorize them, then you can easily communicate with others in the scientific world because you will be speaking the same language. So classification allows orderly grouping of microorganisms. Nomenclature concerns the naming of these organisms. And it's important to note that it requires agreement so that the same name is used by everyone. and therefore it's governed by an international code. And of course, identification, which is able to indicate where the organism should be placed. Okay, so that's what taxonomy is. And for every organism, you should be able to classify it, you should be able to name it, you should be able to identify it. That makes it complete. So this is what we call the 3-domain system by Carl Weiss, which is a system for classifying biological microorganisms based on the differences in their RNA, specifically the 16S ribosomal RNA. It's a structure, that structure hardly changes over time, and so scientists used it as a guide to classify and to group various organisms reliably. And the main purpose for showing you this phylogenetic tree of life is to show you the classification, and that there are 3 domains. You have the bacteria, you have the archaea, and then you have the eukaryota or eukarya, depending on which book you read. Okay, and they are different. Before, bacteria and archaea were placed into one group of prokaryotes, but it was found out based on the ribosomal RNA that they are different. So prokaryote was split into bacteria and archaea. An example of taxonomic ranks used in classification of microorganisms, and of course, I'll use bacteria as an example, include the domain, phylum, class, order, family, genus, species. An example using the organism Escherichia coli, you have the domain, which is bacteria, the phylum, which is Proteobacteria, the class is Gamma Proteobacteria, the order is Enterobacterialis, the family is Enterobacteriaceae, the genus is Escherichia, and finally the species is coli. And so the organism is Escherichia coli or E. coli. And if you notice, it is italicized. The genus is written in capital letters and the organism is written, the species is written in small letters, and you italicize them. And another important thing to note is if you pay attention to the order, it ends with ALES, and the family ends with CEAE. And those again are examples of international codes to use when you're writing organisms. So if I walked into an international conference and I mentioned Enterobacterialis, they will know that I'm speaking about an order. Or if I mention Enterobacteriaceae, they'll know that I'm talking about family. So those are the international codes that govern naming of organisms. So again, taxonomy is very important in microbiology, because once you correctly group organisms, then you will know how to deal with them. You will know how to prevent and control infection, and you will know how to manage and treat infections. For example, you will know when to use an antibiotic, you will know when to use an antiviral or an antifungal. Clearly, if you have identified, named, and classified this organism as a bacterium, there's no way you're going to give an antivirus for it. Or the other way around, if you find out that this is a virus, there's no way you're going to give an antibacterial for it. And so that is very important. And even within the bacteria, it's important to correctly put them in their correct groups, because they're different bacteria that can also be treated and managed differently. And they have different, for example, resistant mechanisms. So if you do not know how to name and classify them, then you will not be able to understand or know how to manage them or know how to approach them. So taxonomy is very, very important. Okay, and so this brings us to the biology of microorganisms. The features of different microorganisms are used to place them in the different taxonomic groups. And how is that? the characteristic features will enable you to properly identify, name, and classify the organisms. And again, I possibly cannot talk about all organisms right now because it's 1 hour. So I will focus on bacteria, the bacteria domain. But it's important to know that even with other organisms, similar processes are used. So the main groups of bacteria are most distinguished by microscopic observation of their morphology and staining reactions. And the most common stain used is the Gram stain. So the Gram staining procedures reflect fundamental differences in cell wall structure, separate most bacteria into 2 great divisions. That is the Gram-positive bacteria and the Gram-negative bacteria. Okay, so this is the main stain that is used as a primary step in identification. And if you notice bacteria, whenever bacteria are spoken of, the first way to describe them is by their Gram stain. Are they Gram-positive or are they Gram-negative? So what is involved in the Gram stain technique? How do we do it in the lab? It has 4 main steps with washing in between the steps. So on a fixed slide, on a fixed stain, you have, on a fixed stain on a glass slide, you have the organism, and this is flooded with crystal violet for 30 to 60 seconds. And crystal violet is the primary stain, and it binds non-covalently to negatively charged molecules in the cell. So once this is washed off, iodine is placed for 30 to 60 seconds, and it acts as a mordant. And what it does is to combine with a crystal violet that is retained by the cells and forms complexes with the crystal violet and the cells. Okay, and then you have, after 30 to 60 seconds, that is washed off, and then you add a decolorizer, which is an alcohol, which is acetone alcohol, and it's added for 15 seconds. And this extracts the complexes, and especially from the Gram-negative. But the complexes, which complexes am I talking about? The iodine crystal violet complex. Okay, so it removes that complex from the cell. And it does that for the Gram-negative bacteria. But for the Gram-positive bacteria, those complexes, the iodine and the crystal violet complexes, are retained by the bacteria, and we shall see why. And then after washing again, another dye, safranine or dilute carboxyl, which is a pink or red dye, is added. And the purpose of this is now to decolorize the unstained Gram-negatives. These are the ones that lost the initial primary stain because of the acetone alcohol. So when you look through a microscope, you have either Gram-positives, which are purple, and that is the color of the first dye. Remember, they did not lose that complex, while the Gram-negatives are pink, excuse me, which is the color of the second dye. And again, so why doesn't the Gram-positive lose the primary stain? Because, and we shall see this in the next couple of slides, is that Gram-positives have a thick cell wall. That is a thick peptidoglycan. Peptidoglycan is the chemical composition of bacterial cell wall. And so the complex that is formed between crystal violet and iodine in the cell is not removed by the decolorizer. But the Gram-negatives have a thin peptidoglycan, a thin cell wall, and so the complexes are easily extracted by the decolorizer. So the cell becomes colorless and then takes up the color of the red dye. So this is what happens in the lab, and hopefully you will get a chance to look at it again in more detail. But the important thing right now is to know why doing a Gram stain is important. And we've already looked at the answer. The Gram stain tells you 2 things. One, it tells you if an organism is Gram-positive or Gram-negative. But another thing it does is it tells you the morphology. Is it rod-shaped or is it cocci? Okay. So by staining the bacterial cell, of course, you're able to determine its morphology. How does it look? Is it round, what we call cocci, or is it in a rod-shaped, what is sometimes called bacilli? Okay, and it's important because these particular shapes give you an idea of the organism that you are dealing with. So if you have gram-positive cocci in clusters, and you talk about gram-positive cocci in clusters in a large gathering, then people will straight ahead think of, oh, they're talking about Staphylococcus, maybe Staph aureus or coagulase negative Staphylococcus. When you talk about gram-positive cocci in chains, then maybe you're talking about Streptococcus. Okay, so it gives you an idea of the organism that you are dealing with. Okay, so two things a gram stain tells you. It tells you if it's gram-positive or gram-negative, but it also tells you if it's cocci or rod-shaped or bacilli. Okay, and there are other gram-negative shapes like spirochetes, comma-shaped for the Vibrio cholerae, the one that causes cholera. Okay, so just having this is important. Knowing this morphology is very important in patient management, because once you give a preliminary report that a clinician has sent in a sample into your lab and you tell them that you're dealing with gram-positive cocci, the medicine they give this patient might be different from the medicine they give somebody with who has gram-negative rods. And so that's why it's important to be able to do the differentiation. And it's important to note also that not all bacteria can be grouped into gram-positive and gram-negative. Some bacteria just don't have a cell wall. For example, the Mycoplasma species, they don't have a cell wall, and so they won't be able to be grouped into any of those gram-positive or gram-negative. And then there's some organisms like the Mycobacterium tuberculosis that cannot be stained with the method of staining using the gram stain, because they have a thick wall which contains mycolic acid. And so the staining, they need another staining technique, which is the ZN technique. I'm not going to go into this right now, but just so that you know that there are other organisms that might not be grouped into gram-positive or gram-negative by virtue of the fact that they cannot be stained using the gram stain. So this is a picture just showing the difference in the cell walls. So for example, you have the peptidoglycan, which is thin. This is the thin peptidoglycan, and we shall look at the cell wall again. So this is the thin peptidoglycan in the gram-negative, and this is the thick peptidoglycan in the gram-positive, okay? And then with the Mycobacterium, you have the peptidoglycan, yes, but you also have mycolic acid. And so this is kind of a hindrance for the gram stain for the mycobacteria. And so you cannot use it to stain mycobacteria. You would need the ZN stain for this, okay? And then of course, fungi can be stained with a gram stain, and it actually looks like gram-positive bacteria, but then it's quite larger. It looks like gram-positive cocci, but it's quite larger than the bacteria, okay? So looking at the structure of the bacteria, that is gram-negative versus gram-positive bacteria. Just checking, Stacy, can you still hear me?
Yes, we can, Dr. Maggie.
Okay, cool, thank you. So looking at the structure of the bacteria, that is gram-negative versus gram-positive, there are differences between gram-positive bacteria and gram-negative bacteria based on their cell envelope. So all this is the cell envelope, and then you have the cell wall, which is the peptidoglycan. In the gram-positive, you can see that the peptidoglycan is thick, and in the gram-negative, the peptidoglycan is thin. Another difference, you can see the gram-negative has an outer membrane. The gram-positive doesn't have it, okay? And this outer membrane of the gram-negative has what we call the lipopolysaccharide. And this lipopolysaccharide is absent in the gram-positive bacteria, okay? The gram-positive bacteria have other structures, which are like teichoic acid and teichoic acid. I'm not going to go into the details of that right now because of time, but just so that you can see that there are differences, okay? And then, so the gram-negative has two membranes. It has a periplasmic space between the peptidoglycan and the inner cell membrane. And then it has the outer membrane, okay? So I'll start with the peptidoglycan. The chemical name, I told you this is the chemical composition of the cell wall. And the peptidoglycan is a very important structure in bacteria. It is strong and relatively rigid, though with some elasticity and openly porous, being freely permeable to some soluble molecules. So it supports the weak cytoplasmic membrane against the high internal osmotic pressure and maintains the characteristic shape of the bacterium. So comparing the gram-positive and the gram-negative, again, the peptidoglycan is thicker in the gram-positives, and that's why gram-positives retain the primary dye in the gram stain. Okay. Something important to note is the basic building block of the bacterial cell peptidoglycan. It consists of two amino sugars, the N-acetylglucosamine and the N-acetylmuramic acid, with a pentapeptide, okay, coming off the N-acetylmuramic acid. Okay, so you have the two sugars and the pentapeptide. And in gram-positives, you have at the lysine part, you have lysine, but gram-negatives, you have what is called the mesodiaminopimelic acid. So that's where a difference between gram-negatives and gram-positives happens. And of course, there are enzymes called transpeptidases, which are important for cross-linking, okay? They're important for cross-linking, for the cross-linkage of the peptidoglycan. And it's very important for you to take note of this structure because it's a target for most of the common antibiotics, which are the beta-lactams, okay? And so the organism tries to, of course, develops resistant mechanisms against beta-lactams that try and break down the cell wall. Okay, and then another important difference I told you is the lipopolysaccharide for the gram-negatives. And this structure is made up of three components. You have the O antigen, which is an O polysaccharide, the core sugars, and of course the lipid A. Something important with the lipopolysaccharide is that it acts as a protective layer for the peptidoglycan, for the gram-negatives. For example, from immune components, from components of the immune system, it impedes the ingress of many antibiotics. And some of the, for example, there's some antibiotics that work for gram-positive organisms, but cannot work for gram-negative organisms because they cannot cross the outer membrane, and simply because of this lipopolysaccharide. So it's important to know that there's some drugs like vancomycin, you cannot give that drug to gram-negatives to infections with gram-negatives because vancomycin cannot cross the outer membrane poring of gram-negative. And the reason it cannot cross because of repulsion by the lipopolysaccharide. Okay, so those are some of the important things to keep in mind when you're thinking about antimicrobial resistance. So it impedes the ingress of many antibiotics, and the lipid A activates the immune system once it is released into blood, and it causes what we call endotoxic shock, which is associated with gram-negative bacteria. Okay. So it's important to note that both the cell membrane of gram-positive and gram-negative And the outer membrane in gram-negatives are associated with proteins, or what we call porins, whose function includes selective permeability, okay? That is the porins. And these are used for intake of nutrients, but antibiotics take advantage of these porins to get into the bacterial cell. And that is why, especially for gram-negatives, as a way of resistance, they lose these porins so that the antibiotics cannot get into the cell. And so you will hear of one resistant mechanism. Oops, somebody's writing on of one resistant mechanism in gram-negatives is loss of the outer membrane porin. So the organism tells itself there's antibiotics, we better lose the way out, the way in for the antibiotic, and they lose the membrane, they lose the outer membrane porin, and so the antibiotics cannot get in, okay? So those are some of the basic structures of bacterial cell, I mean the bacterial envelope, which is one of the most important parts of a bacterial cell. But the bacteria has other cell structures which are important. So some have a capsule, which is important for evasion of the immune system. For example, it prevents phagocytosis by immune cells such as the neutrophils. Then some have flagella, which is important for locomotion. Some have what we call pili and fimbri. This picture doesn't have fimbri, but those are one of the main structures which the bacteria use for genetic exchange between bacteria, but they can also use them for adhesion onto cell surfaces. And then, of course, there are other bacteria that form spores. I do not put a picture in there, but there's spore-forming bacteria which enable them to survive in a dormant state through a long period of starvation or other adverse environment conditions. So moving on to the bacterial growth, physiology, and death. So most of what we know about bacteria derives from their growth. So bacterial growth involves both an increase in the size of the organism and an increase in their number, and you have a net increase in total mass. Bacterial growth and multiplication is of practical value in the detection and identification of pathogens, and it's generally a necessary component of infection. So an organism needs to grow and multiply in order to thrive, okay? And what we understand about bacterial growth is through observations in the lab, and these are summarized in the growth cycle. So we're going to look at that. So the growth curve or cycle represents the number of live cells in a bacterial population over a period of time. And it has 4 distinct phases. It has the lag phase, the log or exponential phase, stationary or decline phase. So the initial phase is the lag phase, where bacteria are metabolically active, but they're not dividing. So this initial phase is characterized by cellular activity, but not growth, okay? So it's important to know them, to know that. The cells increase in size, but no cell division occurs. And then you have the exponential or log phase, and this is a time of exponential growth. It's the rapid growth. So after the lag phase, bacterial cells enter the exponential growth. And this is the time when the cells are dividing by binary fusion and doubling in number after each generation time. There's increased metabolic activity, cell wall components and other substances necessary for growth are generated for division. And it is in this growth phase that antibiotics and disinfectants are most effective, because these substances typically target the cell wall or protein synthesis process of DNA transcription and translation. So at this stage is where the antibiotics and disinfectants would work best. And then the stationary phase, the growth, which is a plateau as the number of dying cells equals the number of dividing cells. So eventually the population growth experienced in the log phase or the exponential phase begins to decline as the available nutrients become depleted and waste products start to accumulate, okay? So the bacterial growth reaches a plateau or a stationary phase where the number of dividing cells equals the number of dying cells, and there's no overall population growth. So the bacterial cells become less metabolic active because the conditions are not favorable. And this is now where the spore-forming bacteria produce endospores in this phase, and pathogenic bacteria begin to generate substances which will help them survive harsh conditions. Okay, and then finally you have the death phase or the decline phase, and it's characterized by an exponential decrease in the number of living cells. So obviously the nutrients become less available, waste products increase, and the number of dying cells continues to rise. So this growth cycle just gives us an idea of the life journey of bacteria. The other growth requirements that are needed for bacteria, which guide us into knowing how the bacteria behave. For example, you have bacteria that are strict aerobes, bacteria that are anaerobes. These ones will not tolerate oxygen. The facultative anaerobes, they grow best aerobically, but can grow anaerobically. The aerotolerant anaerobe, these are anaerobic, but tolerate exposure to oxygen. And you have the microaerophilic organisms. These require or prefers reduced oxygen levels. And finally, the capnophilic, which prefer increased carbon dioxide levels. So just knowing these is very important. For example, if a patient has a deep-seated wound or a deep-seated abscess in the abdomen, then you know that the organism that we are dealing with is a strict anaerobe, which does not tolerate oxygen. And so in the lab, you'll be able to provide this anaerobic condition for this organism in order to grow so that you can study it and do antimicrobial susceptibility tests for it to determine which antibiotic is the best to use. Okay, so it's important to know the growth requirements for, the atmospheric requirements, the oxygen requirements for these bacteria. Some of them require oxygen, some of them don't. Okay, so that's important. And then the same thing can be spoken about for temperature. You have some that grow best at low temperatures, that's the psychrophil. Some that grow best in high temperatures, that's the thermophil. Some that grow best in temperatures between 20 and 40 degrees centigrade, and that's most bacterial pathogens. And that's very important because that's our body temperature is 37 degrees centigrade. So you need to make sure that, it shows you that the organisms that would grow in this particular condition are the mesophils, okay, because our body temperature is 37 degrees centigrade. Okay, and then of course you have other requirements like the pH and the different nutrients and metabolisms that are required for bacteria. So that was bacteria. And again, 1 hour was not enough to cover all these organisms, but I will mention a little bit about viruses. I put an example of the SARS-CoV-2 virus structure, and it's very important to know that viruses consist of a strand, which can be DNA or RNA, that is the nucleic acid genome, and then it's enclosed in a simple protein shell known as a capsid. And some viruses are enclosed further by an envelope, or some are not. And so you have you have enveloped viruses and non-enveloped viruses. And another way to describe viruses is by their nucleic acid genome. Some of them have RNA, some of them have DNA. The SARS-CoV-2 has RNA. And another way of describing viruses is talking about the nature of the nucleic acid genome. Is it single-stranded? Is it double-stranded? That gives you idea of what of which type of virus you're dealing with. And then finally, the fungi. Also, you can have yeast or you can have hyphae, and the yeasts are ovoid or spherical, produced mainly asexually, but some of them can produce sexually. Some of them are dimorphic fungi, being able to grow in different temperature conditions. And then, of course, the molds, which grow as branching hyphal cells. Okay, so that's, I didn't go into much detail about the fungi and the viruses, and also there's protozoa and other parasites, which are important for you to understand. Okay, hopefully you get another opportunity to go through those. Okay, so again, this is the outline that we covered, that we used. I talked about a brief introduction. I told you a little bit about history and taxonomy, and then we dived into the biology of microorganisms, and I mentioned just a little bit about viruses and fungi. Okay, and the references I used to get, for example, the diagrams are placed against the slides, just in case you have an opportunity to get the slides. And then I used the Medical Microbiology by Greenwood, which is a fantastic book if you want to learn the basics of microbiology, especially for undergraduate medical students. Okay, so thank you so much. Thank you for your time.
Okay, thank you very much, Dr. Maggie.
I will just put on my screen so that people can see who is talking.
Okay.
I don't know if you can see it.
Okay. Yes, we can.
Okay.
So thank you very much, Dr. Lubwama. At this point in time, I request anyone with questions or comments, compliments, you can either put up your hand or I will check the chat room for those who have difficulty with network. But yes, please, if you have anything to read now. Yeah. Please unmute. Yes, Dauda.
Okay, thank you very much. Can you hear me?
Yes, we can.
Okay, thank you, Dr. Margaret, for giving us such a wonderful lecture. It was really enlightening. My question is for my team. I'm sure this is almost the same for most teams. We might be having some non-medical oriented students among us. So I'm asking, if possible, will we be getting the slides so we can maybe organize a recap for non-medical oriented students among our team? Thank you.
If I got you right, you're asking that you have non-medical students who might want to review the slides again. Is that what you're asking?
Yeah. So will the slides be shared out?
Yeah, of course. I mean, I will give them to Stacy, and then probably Stacy will and her group will decide. Stacy, are you going to share the slides?
Yes, we are, Dr. Lubwama.
Okay, yeah, so yeah, Zakaria, they will be shared.
Yeah, thank you very much.
Okay, thank you very much, Dauda. Yes, Mohammed.
Hello, thank you, Dr. Margaret, for that wonderful presentation. So my question is on the lipopolysaccharide protective layer of the gram-negative bacteria. So if the gram-negative bacteria has a protective layer, how do drugs then fight these gram-negative bacteria with the protective layer present? Thank you.
Thank you for that question, Mohammed. As I mentioned, and I'll just give one example, but as I mentioned, the organism, the gram-negative organism on its outer membrane has what we call porins. Okay, and those porins are responsible for nutrients getting into the cell, but antibiotics take advantage of those porins and get into the cell. So they get into the cell through what we call the outer membrane porins of the gram-negative. And this is for some of the bacteria. So what happens in resistance is that once the bacteria figures out that antibiotics have started using its outer membrane porin to get in, some bacteria decide to lose those outer membrane porins, and so organisms then fail to get in. But that is in the line of mechanism of resistance. But to answer your question, how do the bacteria get in? Through the outer membrane porin. Somebody would like to know the book that I recommended. It's called The Greenwood by, I'll type it in the chat.
Okay, Mohammed. Yeah.
Stacey, there's someone in the chat who's asking for WhatsApp
Hand
group.
is still up.
That's
the answer. Okay. Yes, Dr. Maggie.
There's somebody
Okay.
asking for a WhatsApp group link that's working.
Yes, I will personally share it at the end of the meeting. But for some of the questions, you've received a lot of thanks, Dr. Lubwama.
Oh, that's nice.
There's a question from there's a question from Idris. Can you please shed more light on endotoxin shock?
Okay, so I didn't really want to get into the detail of this because it will require immunology, which might throw people off. So perhaps but what endotoxic shock is about is the lipid A reacting with some immune cells, leading to the production of cytokines or of some chemicals that activate systems in the blood that lead to seeping out of how can I put it? Dilation of the blood cells, of the blood vessels, sometimes seeping out of the blood. I didn't really want to go into that much detail of it. I probably can put it in the slides before sharing it for those who want to read through it. But for the sake of, I'm not so sure about the depth of immunology right now for the whole group. So I don't think it might be wise to confuse people right now. But I will put a slide before sharing it. Idris, if that is okay with you, I will put a slide about endotoxin shock before sharing the slides, and then you'll be able to read it in there.
Okay, so we have two other questions. Does that difference in peptidoglycan layer size between gram-positive and gram-negative bacteria have some implications when it comes to antimicrobial resistance? And Idris says that it's okay. Yes.
What's the question, Stacey?
So the question is, there's a difference in the peptidoglycan layer size between gram-positive and gram-negative bacteria have some implication when it comes to antimicrobial resistance?
Of course it does, but then it's not just one. Factor alone in the resistance. So when you talk about the peptidoglycan, when you talk about the cell wall, when you talk about the peptidoglycan layer, of course the the gram-positive has a thicker peptidoglycan cell wall. The gram-negative has a thinner one, but remember the outer membrane the outer membrane is actually protecting the peptidoglycan of the gram-negative. So the resistance mechanism in the gram-negative is not necessarily because it has a thin peptidoglycan layer, but because it's being protected by the outer membrane porin. But of course, there's some resistance mechanisms that are similar to gram-positive and gram-negative because of that peptidoglycan layer. And this is where we have the beta-lactamases, which break down the beta-lactams, which act against the cell wall. So it will not, irrespective of if it's gram-negative or gram-positive, the beta-lactam will still act on the cell wall. But the advantage that the gram-negative has is the outer membrane, which protects its peptidoglycan.
Okay, thank you very much, Dr. Margaret. So, ladies and gentlemen, please note that tomorrow we'll be looking at mechanisms. So, more explanations on that will be coming in. Then another question was, thank you for the, oh, sorry, okay. So let me just read it as it is. This is coming in from Ntumba. Thank you for the question, Doctor. The difference in the cell wall composition of other bacteria and M. tuberculosis, do they all have same resistance or drug action mechanisms? So let me read that again. So due to the difference in the cell wall composition of other bacteria and M. tuberculosis, do they all have the same resistance or drug action mechanisms?
So something that's important to note is that these are two different organisms. You have the you have the ordinary bacteria, gram-positive or gram-negative, and you have the MTB. So first of all, the drugs used are different. So the mechanism of resistance will be different and will be based on the action of the drug. So it's not necessarily about the composition of the cell wall necessarily, but the type of drugs that are used in the ordinary bacteria are very different from the type of drugs used in MTB. So the resistant mechanisms will be different. Okay.
Okay, so before we go to the last question, the person who asked the question concerning the difference in sizes of the peptidoglycan layers has said thank you for the elaboration. So last but not least, Adrian, your hand was up. I don't know if you had a question. Okay, so I think he's having a bit of network chat.
His hand is back on. Adrian,
okay.
What's your question?
Thank you very much for the wonderful presentation about microbiology regarding antibacterial growth. I would like to ask the question. So there's bacteria which do not have like a cell wall, okay? I would like to know about about the level of antimicrobial resistance can cause like mycobacterium. Thank you. Mycoplasma.
I'm sorry, I didn't hear the question, Stacey. I missed some of it, maybe my internet.
Are you hearing me?
Now I can hear you, yeah.
Yeah, thank you. My question is that some bacteria don't have cell wall, okay? For example, Mycoplasma. How about if regard compared to the antimicrobial resistance for those which does have a cell wall and other don't have cell wall? How about degree
Okay,
for
so
antimicrobial resistance can contribute?
okay.
Thank
That's
you.
a good question, because what that question is saying is, remember, antimicrobial resistance, you have to know the drug being used, and you have to know the mechanism of action of the drug. And from that, you will then discover that, okay, what does the organism do to try and stop this antibiotic? So Mycoplasma doesn't have a cell wall, and therefore you will not use the ordinary drugs that you would use, like the beta lactams, which act on the cell wall. So the mechanism of resistance of the drugs for Mycoplasma are the drugs that, for example, act within the nucleic acid. For example, the fluoroquinolones, the what is this called? The erythromycins, the macrolides, etc., etc. So that governs the resistance that you will see in Mycoplasma, the type of drug. So that's something that you should always have in mind when you're trying to compare these resistant mechanisms. Look at the two organisms and then look at what drugs are used for these organisms. And then what is the mechanism of action of these drugs? And after answering that question, then you go upward and say, what does the organism do to try and stop this drug from working? Okay, so the Mycoplasma has different drugs compared to the other ones. And of course, the other ordinary bacteria with cell walls can also, the other drugs can work with them, but it's just that Mycoplasma, because it doesn't have a cell wall, will not have, you cannot use bacteria against cell walls. I mean, you cannot use antibiotics against cell walls for the Mycoplasma. Does that make sense, Adrian?
Thank you very much. I understand
Okay.
now. Yeah.
Okay.
Perfect.
Okay, so thank you so much, Stacey, for having me give this class.
Thank you very much, Dr. Lubwama. I don't know if, okay, I think that was, it. I'm seeing more thank yous and then a request for the group link, which is about to be shared. So thank you very much, Dr. Lubwama, for sparing time and coming to give an elaboration on the biology of microorganisms. Yes, so at this point in time, I'm going to share the group link. I've just done it. Please note that this link won't really be active for much longer, so join in before it becomes inactive. Yes. So thank you very much, everyone, for attending. This is the end of the session. The presentation will be shared. Yes. Okay.
Okay, thank you. Have a good evening.
Have a good evening, Dr. Lubwama. So I'm going to give probably just 3 more minutes for those who haven't been able to join in the WhatsApp group. The link is there. Yeah. Adrian, your hand is still up. Is it for the previous question you had asked, or? Okay. What I'd like to make mention of, please, when you join in,